Hello everyone, welcome to our YouTube channel. Today, our focus will be on identifying disease-causing mutations through bioinformatics and computational approaches. Today, we will cover its overview, importance, recent trends, tools, project ideas with a final note. Let's get started. So, identifying disease-causing mutations through bioinformatics and computational approaches is a critical area of research in genetics and bioinformatics. This process not only aids in understanding the genetic basis of diseases, but also plays a vital role in developing targeted therapies and personalized medicines. So, what is the importance of studying disease-causing mutations? Studying disease-causing mutation is essential for several reasons, like understanding disease mechanism. Identifying mutations helps elucidate the biological mechanism underlying various diseases, particularly rare genetic disorders. Then, personalized medicine. Knowledge of specific mutations allow tailored treatments based on individual genetic profile, improving therapeutic efficacy. And public health. Understanding genetic predispositions can inform public health strategies and genetic counseling, potentially reducing the incidence of hereditary diseases. Let's discuss some recent trends in computational approaches. Recent advancement in computational biology and bioinformatics have significantly enhanced the ability to identify disease-causing mutations. Key trends include next-generation sequencing. Next-generation sequencing technologies have revolutionized genomics, allowing for the rapid sequencing of entire genomes and the identification of mutations associated with diseases. Machine learning. The application of machine learning algorithms to genomic data has improved the accuracy of mutations, predictions, and classification, facilitating the identification of pathogenic variants. Privacy-preserving techniques. New methods are being developed to analyze genomic data while ensuring patients' privacy, utilizing secure multi-party computation to protect sensitive information during mutation analysis. Now, let's go through some powerful bioinformatic approaches and tools. Here are some of the most common bioinformatic tools used to identify disease-causing mutation, like GATK, Genome Analysis Toolkit. Its purpose is primarily used for variant discovery and genotyping from next-generation sequencing data. It has features like it provides a comprehensive pipeline for processing NGS data, including quality control, variant calling, and annotations. Then, Variant Effect Predictor. Purpose: annotates variants to predict their functional consequences. Features: integrates data from various sources to produce insights into the potential impact of variant on gene function and disease association. Then, ANNOVAR. Purpose: it annotates genetic variants detected from sequencing data. Features: it offers a flexible and comprehensive system for the functional annotation of variants, allowing researchers to filter and prioritize variants based on various criteria. Then, SIFT, sorting intolerance from tolerant. Its purpose are: it predicts whether an amino acid substitutes affect protein function or not. Features: uses sequence homology and the physical properties of amino acids to assess the potential impact of mutations. Then, Polyphen, polymorphism phenotyping version 2. Purpose: it predicts the possible impact of an amino acid's substitution on the structure and function of human protein. Features: it provides a score indicating like the variant is damaging based on multiple sequencing alignments and structural information. Then, 5. Purpose: ranks variants based on their similarity to known diseases phenotypes. Features: it utilizes data from knockout experiments in model organisms to inform variant. Then, clean variants. It's a public database that archives relationships among genetic variants and phenotype. Features: it provides information on the clinical significance of variants, aiding in the identification of disease-causing mutation. Then, human phenotype ontology. Purpose: it provides a standardized vocabulary for describing phenotypic abnormalities associated with human diseases. Features: it facilitates the association of clinical features with known diseases, genes, aiding in variant prioritization. Then, VAR sequence. Purpose: a commercial software tool for variant interpretation and prioritization in a clinical context. Features: it offers user-friendly interface for geneticists and clinicists to analyze and report variants. Lastly, Ngenuity variant analysis. Purpose: a commercial tool for analyzing genetic variants in the context of disease. Feature: it integrates various biological data sources to assist in the interpretation of variants and their potential clinical significance. These tools are essential in the bioinformatics pipeline for identifying and analyzing disease-causing mutations, especially in the context of next-generation sequencing data. They help researchers filter, annotate, and prioritize variants, ultimately contributing to the understanding of genetic disease and the development of targeted therapies. So, here are some research project ideas. Here are 4 detailed research project ideas focused on the identification and validation of disease-causing mutations, including objectives, step-by-step processes, and the tools required for each project. Project 1: Machine Learning for Variant Classification. Objective: developed a machine learning model to classify genetic variants as pathogenic or benign disease. Key steps involve data acquisition. Source: obtain a dataset's known variants from reputable public databases. Content: ensure the data includes variant identifiers, genomic locations, associated diseases, and classification. Second step: data preparation. Feature extraction: extract relevant features like evolutionary conservation, physiochemical properties, and functional predictions. Data cleaning: handle missing values, remove duplicates, and standardize data formats. Then, model selection. Algorithmic choice: select suitable machine learning algorithms for classification tasks. Library: utilize libraries like scikit-learn for implementations. Model training. Data splitting: divide the datasets into training and testing sets. Then, optimize model parameters for optimal performance. Model fitting: train the model on the training data. Then, model evaluation. Matrices: assess model performance using various matrices. Confusion matrix: visualize model performance. Then, validation. Test the dataset. Validate the model on the reserved test dataset. External validation: if possible, validate on an independent dataset. Deployment: web interface. Create a user-friendly interface for input and output. Documentation: provide clear instruction and limitations. So, the tools required here are: programming language, Python. Libraries, scikit-learn, pandas, numpy, matplotlib for visualization. Second project: identification of disease-causing mutation using next-generation sequencing. Objective: to identify and validate pathogenic mutations in patients and rare genetic disorders using next-generation sequencing data. Detailed steps include patient requirement, recruit patients with suspected genetic disorder and obtain informed consent of genetic testing. Sample collection. Collect blood or saliva samples from patients for DNA extraction. Third, next generation sequencing. Perform whole exome or whole genome sequencing on the extracted DNA using platforms like Illumina or Thermo Fisher. Then, data control. Use FastQC to assess the quality of the raw sequencing data. Trim low-quality reads using Bioinformatics. Then, variant calling. Align the clean reads to a reference genome using BWA or bowtie. Then, call variants using GATK or SAM tools to generate list of SNPs and indels. Then, variant annotations. Annotate variants using ANOVAR or VEP to determine their potential impact on gene function. Then, filtering parameterization. Filter variants based on allele frequency using database like Genome80, predicted pathogenicity using tools like SIFT and Polyphen, and an inheritance pattern. Prioritize variants that are likely to be causative based on the patient's phenotypes and family history. Then, validation. Validate candidates' mutation using Sanger sequencing or another methods to confirm their presence and the patient. Clinical reporting. Prepare a report detailing the identified mutations, their potential clinical significance, and recommendations for further testing or management. Required tools for this are NGS platforms, Illumina, and Thermo Fisher. Bioinformatic tools are FastQC, Thermomatic, BWA, GATK, SAM tools, ANOVAR, VEP, SIFT. Third project is on machine learning for predicting pathogenicity of variants. Objective: to develop a machine learning model that predicts the pathogenicity of genetic variants based on genomic features. Detailed features are data collection. Gather a dataset of known variants like databases like CleanVar or GenomeEd, including their classifications. Data processing. Clean the datasets by removing duplicates and handling missing values. Extract relevant features such as conservation scores. Use tools like FileLop. Function predictions utilize SIFT and Polyphen. Features engineering. Generate additional features based on data such as mutation types, missense and nonsense, genes expression levels, and allele frequency. Model selection. Choose machine learning algorithms for classifications such as random forest, support vector machines, SVM, or gradient boosting. Then, model training. Split the datasets into training and testing sets. Train the model on the training set and tune hyperparameters using cross-validation. Model evaluation. Evaluate the performance using matrices such as accuracy, precision, recall, and F-word score on the test set. Use a confusion matrix to visualize classification results. Validation. Validate the model with an independent dataset to ensure its generalizability. Deployment. Create user-friendly interface for clinicals to input variants and receive predictions. Tools required are for programming language, Python, libraries, scikit-learn, pandas, numpy, matplot library for visualization, machine learning frameworks, TensorFlow, or Keras for deep learning models, if applicable. Fourth project idea is on privacy-preserving analysis of genomic data. Objective: to develop a framework for identifying disease-causing mutations while ensuring patients' privacy through secure multi-party computation. Detailed steps include literature review, Conduct a literature review on existing privacy-preserving techniques in genomic data analysis. Framework design. Design a secure multi-party computation protocol to analyze genomic data without revealing individual patient information. Data acquisition. Collaborate with hospitals or research institutions to obtain genomic data for analysis while ensuring compliance with ethical standards. Prototype implementation. Implement with the SMPC protocol to perform analysis on the genomic data, ensuring that only the results are disclosed. Testing and validation. Test the framework on simulated genomic data to evaluate its performance and ensure it meets privacy and requirements. Validate the framework using real genomic datasets while maintaining patients' confidentiality. Performance evaluation. Evaluate the performance of the protocol based on execution time, scalability, and resource consumption. Documentation and reporting. Document the framework, including its design, implementations, and evaluation results. Prepare a report detailing the identified mutations, their potential clinical significance, and the privacy measures employed. Tools required: programming languages, Python or C++. Libraries, cryptographic libraries, specific SMPC frameworks. These projects encompasses a range methodologies for identifying and validating disease-causing mutations, from experimental approaches using NGS to computational techniques involving machine learning and privacy-preserving frameworks. Each project emphasizes the integration of biological data with computational tools, fostering a deeper understanding of genetic diseases and their implications for patient care. Join our project and training sessions to delve deeper into the world of computational genomics. Like, subscribe, and comment for more insights and update on our research initiatives. Together, we can advance the understanding of genetic diseases and improve patient outcomes. Thank you all.